The aim of this procedure is to express functional antigen-specific T-cell receptors on mouse T-cells. First, transfect a retroviral-producing cell line with plasmid containing the TCR gene of interest to package TCR-expressing retrovirus. Next, isolate and purify T-cells from mouse spleens. Infect these purified T-cells with the retrovirus produced in the first step. Finally, expand the transduced T-cells to express and obtain appreciable levels of receptor. Ultimately, results can be obtained that show the expression of functional antigen-specific TCRs on mouse T-cells through flow cytometry.
Hi, my name is Michelle Kroesgaard. I'm at the NYU Cancer Institute at NYU Medical School. Today we're going to show you how to transduce primary T-cells. The people who will be showing you how to do this will be Shi Song, a postdoc in the lab, Carolina Melchek, a graduate student in the lab, and Ariane Perez-Garcia, a postdoc in the lab.
The main advantage of this technique over existing methods like transgenic mice is that it is much less time-consuming.
This method can help answer key immunological questions such as which TCR can give better response against antigens.
This technique can be applied for adoptive T-cell transfer therapy, which has been shown to be promising for the treatment of cancer patients and consists in the in vitro modification of human T-cells.
To ensure equal expression of the T-cell receptor alpha and beta chains, subclone the gene of interest into the same retroviral vector under control of the same promoter. Prepare high-quality plasmid DNA using the QIAGEN Maxi Prep Kit and make a stock of 1 microgram per microliter. Remove media from the plate with platinum E retroviral packaging cells and wash the plate once with 1x PBS. Dislodge the cells by adding trypsin EDTA and incubate at 37 degrees Celsius for a few minutes. Neutralize the dislodged cells with DMEM media and transfer to a falcon tube. Next, centrifuge the cells at 1000 times G for 5 minutes. Aspirate the supernatant and resuspend the cell pellet in DMEM media. Determine the cell number and dilute the cells at 0.6 times 10 to the 6 per milliliter. Plate 10 milliliters of cell suspension in a 10 mm polylysine coated tissue culture plate and grow overnight in a cell incubator at 37 degrees Celsius in 5% carbon dioxide. The next morning, examine the cells under a light microscope to verify approximately 80% confluency. Gently remove the media from the plate, wash the cells once with 1x PBS. And add 10 milliliters of pre-warmed fresh DMEM media without penicillin or streptomycin. For the lipofectamine transfection complex, prepare 2 mixes in OptiMEM, one of DNA and the other of lipofectamine reagent. Equilibrate separately for 5 minutes at room temperature, then mix together gently and incubate for 20 minutes at room temperature. Slowly drip the mixture into the platinum E cells. Gently rock the plate back and forth to distribute the transfection mixture evenly. Then incubate cells at 37 degrees Celsius and 5% carbon dioxide in a cell incubator. After 6 to 8 hours, replace medium with 10 milliliters of fresh DMEM medium for viral production. Harvest a mouse spleen and transfer to RPMI medium. Place the spleen in a cell strainer. Mash the spleen with a syringe plunger into a 5 cm tissue culture plate. Rinse cells off the cell strainer with sterile PBS. In order to harvest the cells, centrifuge at 1000 times G for 5 minutes. Discard the supernatant. Continue by resuspending the cell pellet in ACK lysing buffer, adding 2 milliliters per spleen. Incubate for 2 minutes at room temperature. Add RPMI medium up to 20 milliliters and centrifuge at 1000 times G for 5 minutes. Finally, resuspend the cell pellet in sterile PBS. Determine the cell number and centrifuge at 1000 times G for 5 minutes at 4 degrees Celsius. In order to activate the mouse T-cells before viral transduction, coat plates with activating antibodies, namely anti-CD3 epsilon and anti-CD28. Prepare an antibody mixture of 1 microgram per milliliter of anti-CD3 epsilon and 2 micrograms per milliliter of anti-CD28 in sterile PBS. Dispense 250 microliters of the antibody mixture into each well of a 24-well tissue culture plate and incubate for 2 hours at 37 degrees Celsius. Magnetically label the spleen cells according to the product manual. Place an LS column in the magnetic field. Apply labeled cell suspension onto the column. Collect flow-through of enriched mouse CD8 plus T cells. Wash the column 3 times with 3 milliliters of buffer and combine with the previous flow-through. Stain the cells with anti-CD3 epsilon and anti-CD8 alpha antibodies and analyze by flow cytometry. Next, centrifuge the cells at 1000 times G for 5 minutes and resuspend the cell pellet to 1 million cells per milliliter in rPMI medium with recombinant human IL-2. Remove the antibody solution from the activation plate. Rinse each well with sterile PBS. Add 1 milliliter of the cell suspension to each well and place the plate at 37 degrees Celsius 5% carbon dioxide in a cell incubator. After 2 days of virus production, the medium in the platinum E cell plate should become yellow. Transfer the viral supernatant to a 15 milliliter conical tube. To remove cell debris, centrifuge the virus supernatant at 1000 times G for 5 minutes. Carefully transfer the virus supernatant to a new tube. Leave some liquid at the bottom of the tube and do not disturb the cell's debris. Collect activated T cells from the plate into a 50 milliliter conical tube. Determine the cell number. Save some cells in a separate tube to be used as a negative control. Centrifuge at 1000 times G for 5 minutes and discard the supernatant. Then, resuspend the cell pellet in virus supernatant at 10 to the 6 cells per milliliter with 20 nanograms per milliliter of recombinant human interleukin-2 and 10 micrograms per milliliter of protamine sulfate. Add 1 milliliter of cell suspension to each well of a 24-well plate. For the control tube, resuspend the cells in rPMI medium at the same cell density with the same amount of recombinant human interleukin-2 and protamine sulfate. Add the cells to the same plate. Wrap the plate with plastic film. Centrifuge for 90 minutes at 2000 times G at 32 degrees Celsius with no break. After the centrifugation, remove the plastic film. Add 1 milliliter of fresh rPMI medium with 20 nanograms per milliliter of recombinant human interleukin-2 to each well. Put the plate back into the incubator. It is important to examine the transduced T cells daily. Split the cells at 1 to 3 ratios when necessary. Do not let the cells overgrow or let the medium turn yellow. At day 6 or day 7, stain the cells with antibody and MHC tetramer specific for the TCR to evaluate the expression of TCR on T cell surface. Use the untransduced T cells as a control. These transduced T cells are now ready for further downstream applications. Prior to viral transduction, it is advantageous to obtain high-purity T cell subsets using commercial magnetic beads. To evaluate the expression level of TCRs on T cells, antibodies specific for TCR alpha or beta chains can be used. Typically, 30% to 80% of the cells can express transduced TCR, depending on the TCR genes and virus titers. MHC tetramer specific for the TCR can also be used to further verify functional expression of TCRs.
Once mastered, this technique can be done in 1 week if performed properly.
While attempting this procedure, it is important to remember to keep the cells in healthy condition.
Following this procedure, other methods like cytotoxicity assays or cytokine release assays can be performed in order to answer additional questions like the specificity and the sensitivity of the transduced TCRs.
After watching this video, you should be an expert in transducing your favorite TCR into primary T cells. So, see you in the lab.